Hello, I'm Chris Fry. I am a group leader at Cell Signaling Technology, and it's my pleasure to share with you the story of an antibody. This is a tale of one of CST Antibody's conception, development, production, and release for use in biomedical research. You may be surprised to learn that here at CST we do things a bit differently. First, we develop and produce all of our primary antibodies in-house. We also perform all validations in-house, testing for antibody specificity, sensitivity, and cross-reactivity in multiple applications. And if you need technical support, the same CST scientists who produced and validated your antibody will personally answer your call or email, sharing their experience and their data. We'll focus on a single antibody, but the story is just one example of how all rabbit monoclonal antibodies are developed at CST. Let's start at the beginning, with our recognition of the scientific community's need for a marker of cancer progression. Dr. Jing Li, one of our development scientists, met with a committee of CST scientists, including cancer researchers, actively publishing in high-impact journals. They identified the need for a highly specific and multi-purpose antibody targeting vimenin, a protein associated with the epithelial to mesenchymal transition. This transition from adherent epithelial cells to migratory mesenchymal cells is required for some normal developmental processes, but it also marks the initiation of metastasis and cancer progression. Dr. Li was tasked with developing a vimenin antibody for use with fluorescent and colorimetric cell and tissue imaging, and whose specificity could be verified by Western blotting. To get the project started, CST scientists specializing in bioinformatics and molecular modeling designed antigenic peptides to elicit a strong immune response in rabbits. Peptides were synthesized by our in-house peptide lab, and then rabbits were immunized and screened by Dr. Li for reactivity and specificity for vimenin. Go ahead, rotate the 3D molecular model of vimenin. Dr. Li requested a clonal expansion of antibody-expressing cells from the best rabbit. Using proprietary technology that allows access to a broader range of antigen-expressing cells than traditional methods, the CST monoclonal development group created nearly 1,000 clones. They then screened them by ELISA, passing only those with the most robust signal back to Dr. Li for analysis. Screening such a large number of clones increases the chance of identifying a really outstanding antibody. Dr. Li performed many Western blots to further assess the specificity of these antibodies for the vimenin protein. Some of this data is available for you to explore at the bottom of your screen. First, the initial 71 clones identified by ELISA screening were evaluated using COS7 cell extract. Of these, 21 demonstrated a specific band at the expected molecular weight for vimenin. Dr. Li then went on to test the antibody specificity of these 21 clones by Western blot in cell lines from other species including human, mouse, rat, and monkey. Dr. Li also submitted 21 clones for screening by our immunohistochemistry and immunocytochemistry groups. Vimenin forms intermediate filaments of the cytoskeleton and is present in the cytoplasm of mesenchymal cells. IHC analysis identified some clones that demonstrated only weak cytoplasmic staining on HeLa cells and weak mesenchymal staining on breast carcinoma tissue. Although these clones stained cells and tissues as expected, they were dropped from subsequent testing due to weak signal strength. The remaining clones were further tested on breast carcinoma tissue sections and as a control on BT474 cells, which do not express vimenin. Of these remaining clones, only 4 showed robust, clean, and specific staining. Using fluorescent immunocytochemistry on cultured COS7 cells, we also verified that immunostaining was specific to intermediate filaments. The high level of rigor we apply to antibody screening and validation is possible only because of the integrated efforts of multiple dedicated teams of scientists at CST. You can use the same optimized and standardized protocols that we use and be confident of getting reproducible results from experiment to experiment and from lot to lot. Dr. Li assessed the performance of each clone in the multiple applications that were tested and chose the most specific and robust clone for production. The antibody was formulated based on its performance in each of these applications and then submitted to our immunohistochemistry and cytometry groups for the determination of optimal dilution for IHC, IF, and flow. In addition, the fluorescent immunocytochemical staining of the vimenin antibody was compared to 2 competitor antibodies. As you can see from the data, The new CST antibody outperformed one of the competitors and performed equally as well as the other. This clone qualified for distinction as a CST-XP antibody on the basis of its exceptional performance and utility for multiple applications. At CST, having one group of scientists achieve specific results with an antibody isn't enough. We take the extra step of having in-house production scientists conduct further tests to ensure our customers can achieve reliable and consistent results in their labs. In the case of vimenin, CST production scientist Julie Diamond repeated the Western blot analysis, confirming the antibody specificity seen by Dr. Lee. Julie also observed that the CST vimenin antibody outperformed 3 competitor antibodies, which displayed weaker signal and nonspecific banding with longer exposure. Julie is a member of Production Team 3, which is responsible not only for production, but also for providing technical support for all vimenin antibodies. Having worked with this antibody personally, she is in a unique position to provide customers with knowledgeable and helpful support. As we do for all of our primary antibodies, Dr. Lee tested the vimenin antibody's stability and activity by Western blot following a storage period of 7 days at room temperature and 37 degrees Celsius. The performance of this antibody was not compromised by storage at high temperature, so we know the antibody can be shipped at ambient temperature. This enables us to reduce the environmental impact of unnecessary packaging. But we're not done yet. Members of each of the CST scientist teams that tested and optimized the antibody must sign the antibody release form, underscoring their personal commitment to the product's quality and performance. However, the best validation of this product has come from the many researchers who have used it and published their results. This CST vimenin XP antibody has now been cited in more than 45 peer-reviewed publications since its release in November of 2010. Thank you for viewing the story of an antibody. We encourage you to search our website for a thoroughly validated antibody targeting your research. This vimenin antibody story is one of many stories at CST from a line of products that is far more extensive than many scientists are aware of. Cell signaling technology strives to be a valuable partner at your bench. That's why every product goes through the same rigorous testing and validation process. We do this because that's what we would want from an antibody if we were in the lab. Because actually, we are.